Hello, everyone. Welcome to another TPD seminar. Today, it's my greatest pleasure to introduce our speaker, Dr. Stephen Banik, who is an assistant professor in the Department of Chemistry and a ChemH Institute scholar at Stanford University. He received his BS from University of Wisconsin and his PhD in chemistry from Harvard University, working with Dr. Eric Jacobson. He performed his postdoctoral studies with Dr. Carolyn Bertozzi at Stanford University, where he developed Litex, which you're going to hear about today. Stephen began his independent career in January of 2021. At Stanford, Stephen's group combines chemical biology, molecular biology, genomics, proteomics, and protein engineering for translational tool development and the study of biological mechanisms. Very excited to have you here today, Stephen, and we're looking forward to hearing about your work in your lab.
Great. Thank you so much, Catherine. And thanks to the entire organizing committee for the invitation. This is a really wonderful sort of service you provide to the community. And I've certainly enjoyed sort of all the speakers during this webinar series. And I'm excited to kind of share a little bit about how I think about targeted protein degradation, maybe from a space that's a little bit different from how other speakers have talked about in the past, and that's thinking about it from the other side of the cell. How can you degrade a protein from the extracellular space? And I think this idea really comes from a place of thinking about how we understand molecular machines inside a cell. So for a long time, we've understood proteins and protein function through the lens of stopping that function. So asking a question by preventing a function and seeing what happens after that. And this has been incredibly transformative. This has allowed us to develop inhibitors. This has allowed us to really just nail down a bunch of interesting mechanistic biology in a number of different therapeutic contexts. I think a different but no less exciting question is, rather than stopping a protein's function, what if you ask that protein to do something else? What if you ask that protein to perform a new function that it wasn't performing before, or to ask it to do something outside its normal scope? And so I think that question is really sort of a broader question, asking what is the potential scope of biology? And that is incredibly exciting. And I think as we've already learned, is incredibly transformative for new therapeutic opportunities. And it's not really a new idea. The discovery of small molecule molecular glues that allow us to glue two proteins together, like FK506 and its ability to bring together calcineurin and FKBP12, really sort of instigated this idea that reprogramming a protein's function with some sort of exogenous reagent or exogenous molecule could allow us to tap into some new features of a mammalian system. And I think more recently, that same sort of theme has cropped up in a number of different contexts. And one that's incredibly exciting, I think, is in our ability to manipulate the genome selectively, where we can use an exogenous reagent, in this case, a Crispr protein, to perform site selective chemistry inside a cell using the machinery of mammalian cells. And so this idea of asking pieces of a mammalian cell to do something new, I think, is extremely exciting. And conceptually, I think one of the biggest growth areas for this, based on the seminar series, is targeted protein degradation. And I think as a conceptual technology, this area has a large number of advances, which I don't need to convince this audience of. But where I see sort of major conceptual leaps of targeted protein degradation is in our ability to remove a protein without necessarily knowing its function, without having a discrete binding site. So rather than trying to develop a specific molecule that stops a specific function in a specific place, we might have a little bit more room to breathe in terms of developing molecules that can get rid of that protein. Many proteins perform multiple different functions, and not all of them can be blocked by the same reagent. And so removing a protein provides us an avenue to get rid of the secondary functions of a protein, or scaffolding functions of a protein. And then a context that I think about a lot is the context of our ability to inhibit some sort of ligand protein interaction. And this is a complex equilibrium. Whenever we develop an inhibitor for some function, it's always an on-off. And so depending on how sensitive the threshold is for stimulation of your protein by some ligand, you might not be able to reach an effective inhibitory concentration that allows you to prevent all of this activity. And so by lowering the concentration of that protein, you can bypass this limitation in terms of this on-off equilibrium. And of course, there have been a number of exciting developments in the area of intracellular protein degradation, or degrading proteins using the machinery that is inside the cytoplasm of cells. And so molecules like protacs and molecular glues are sort of in this area of small molecules that impart some sort of effector function on a target of interest, or redirect cellular effector function to a target of interest. Often this is ubiquitination and then downstream degradation in the proteasome, but there have been exciting recent examples where this is also some other post-translational modification editing activity. A separate class of molecules that I think is also incredibly exciting is our ability to maybe not couple an effector function to a target protein, but to manipulate the interactome of that target by bringing two proteins together that weren't interacting at a high enough frequency and removing that target protein through that interaction. And so examples of small molecules that take advantage of intracellular autophagy pathways like AUTAX and ATEX are also incredibly exciting developments in the area of targeted protein degradation. But all these intracellular technologies rely on our ability to develop a ligand for an intracellular protein, and rely on our ability to tap into the intracellular protein degradation machinery. And not all targets satisfy the requirements for those different criteria. And so this is where I, in my postdoctoral work, really got interested in asking the question, just as we have very powerful techniques for intracellular protein degradation, could we develop similarly powerful technologies for degrading extracellular proteins, or degrading proteins from the other side of the plasma membrane? And I think this is incredibly important because many of the most important proteins out there in terms of disease context exist in the extracellular space. These are proteins that drive cell-cell interactions, that drive cell environment interactions, that dictate when a cell is going to implant in a tumor microenvironment, for example. These are the proteins that send and receive signals. This is how cells talk to each other throughout the body. And certainly many of these pathways are co-opted in many different disease states, including cancer. And then the secreted proteome represents a lot of the effectors of the immune system of our bodies. So in diseases, these can become things like protein aggregates, but there are also antibodies, tissue remodeling enzymes, complement factors, a number of proteins that drive diseases in a number of different contexts. And so I wanted to develop a targeted protein degradation strategy that might be able to more readily access this scope of targets. And that's where we asked a really fundamental question, which was, how can we take advantage of removing a protein from the extracellular space? And the endosomal lysosomal pathway is a really attractive pathway to take advantage of for this purpose. And the reason is because this is what is naturally degrading the scope of proteins in our cells. And so what we sort of hit on was the idea that we might be able to couple the activity of specific receptors that drive cargo to lysosomes with the degradation of a target protein. And so the really fundamental question here was, can we use the trafficking activity of a lysosome targeting receptor to remove another protein that might be a deleterious protein in a disease context? So you could do this for both secreted proteins as well as for membrane proteins. And specifically in the context of membrane proteins, this would be incredibly exciting because it asks, again, another fundamental question. When you pair two proteins together, which protein's activity is going to dominate the other one? And that's, I think, a new lens to think about the biology of these lysosome targeting receptors, as well as the targets that we might go after to degrade. And the key to this strategy would be to develop a bifunctional molecule that allows us to bridge a lysosome targeting receptor with a target protein of interest. And so when we started this, there wasn't a lot known necessarily about specific lysosome targeting receptors, but there were some very concrete, clear examples. And so one of the most historically well-characterized receptors is the cation-independent mannose-6-phosphate receptor, whose job is to naturally drive cargo from the Golgi to lysosome. So a number of our lysosomal hydrolases that go to the lysosome are driven by the mannose-6-phosphate glycan mark present and somewhere in their protein structure. And it also exists to capture cargo from the extracellular space. And so we thought that if we could hijack the mannose-6-phosphate receptor, we would have a good chance of at least showing this idea that a lysosome targeting receptor could be co-opted for degrading a target protein. And so the design principles for developing a molecule that could do this, or a lysosome targeting chimera, are, I think, somewhat less confined than when you think about the intracellular space. So in its simplest form, for a LITAC molecule, all you need is a target binder and some sort of ligand for your lysosome targeting receptor. And our initial iterations, this was the cation-independent mannose-6-phosphate receptor. And so we imagined that you could take anything from an antibody to an antibody fragment to an aptamer, all the way down to a small molecule, and use that as a target binder. And that if you link these in a sort of easily accessible or efficacious way, this would allow us to readily assemble binder ligand pairs. And so our initial molecules used a relatively complex 15-step synthetic route for a number of glycopolymers. We've since moved to simpler structures to sort of make these more accessible. And then finally, the ligand for the receptor that you're targeting is incredibly important. And so because we chose the cation-independent mannose-6-phosphate receptor, there was literature data out there that told us what the design requirements might be for these ligands. And so we chose to assemble some hydrolytically stable mannose-6-phosphate variants called mannose-6-phosphonate, and to assemble these in a multivalent manner on the backbone of our glycopolypeptides, because sort of this multivalent presentation of ligands is important for driving the cation-independent mannose-6-phosphate receptor to deposit cargo in lysosomes. And so we assembled some very early proof of principle molecules for this. So we just took biotin, a small molecule, and assembled a bifunctional LITAC molecule using our mannose-6-phosphonate glycopolypeptides. And we asked, when we took cells with a biotin binding protein present in cell culture, would we now drive that protein to lysosomes in cells using a biotin LITAC? And so we made a number of control molecules, and really the takeaway was that yes, we could indeed see uptake of a fluorescently labeled biotin binding protein into cells when we use the biotin LITACs, especially in comparison to control glycopolypeptides like polygalnac or polymannose. And that this uptake activity was inhibited in the presence of a large excess of monomeric mannose-6-phosphate, suggesting that there was some sort of specificity in this uptake pathway. But we really wanted to know, where was the protein going? Where was this Nutravidin 647 going when we were bringing it into cells? And could we track this progress over time? And so we used confocal microscopy, and we quickly saw that even within 1 hour of exposure to a LITAC molecule, fluorescently labeled protein was being driven to lysosomes very quickly. And then we could track this over time, both by fluorescence as well as in-gel fluorescence. And we could see that these uptake kinetics were relatively consistent with a process where the rate limiting step was recycling of a proposed receptor back to the cell surface. And so at this point, we really wanted to, I think, understand the potential scope of cells that might be accessible for this strategy, because in principle, all cells with lysosomes have the mannose-6-phosphate receptor, because it's the primary way that cargo is delivered to lysosomes from intracellular spaces, from the Golgi. And so in a small screen of cell lines, we observed that biotin litacs that target the mannose-6-phosphate receptor were indeed able to bring in fluorescent proteins into a number of different cell lines. I think sort of suggesting and speaking to the broad presentation of the mannose-6-phosphate receptor across mammalian cells. But we wanted to understand a little bit more about maybe what are the cellular features that would be important for driving this process. So if we wanted to develop a therapeutic approach, we need to sort of understand the cellular mechanisms that are going to define whether or not we could bring a protein into a cell and degrade it. And so to ask that question, we turned to sort of a genome-wide Crispr knockdown screen to assess maybe what are the cellular requirements for effective uptake through the mannose-6-phosphate receptor pathway. And what we did was we took K562 cells that stably expressed the CRISPR-I machinery, so dCas9 fused to CRAB. We generated a sort of a genome-wide knockdown library across the whole genome, so about 20,000 proteins or 200,000 guides. Then we exposed this cell library to a litac, and we performed our uptake experiments. And then we sorted on the sort of lower 10% of cells that did not perform effective uptake, and then looked at what genes might be responsible for regulating the internalization of litac molecules. And when we did that, sort of gratifyingly, we got back the mannose-6-phosphate receptor. And so I think when you do a genome-wide screen for uptake through a specific pathway and you get the receptor that you're targeting very specifically, you feel pretty good about the design of your ligand and the design of your approach. And furthermore, we didn't really see any other endocytic receptors. So we knew that we were hitting the mannose-6-phosphate receptor exclusively. But we also discovered a number of other interesting hits as well from the screen. And so I'll talk a little bit more about the exocyst complex, which is highlighted here in orange. But more generally, these black dots that we returned from our genome-wide screen really allowed us to map the whole endolysosomal pathway as being important for effective uptake through the mannose-6-phosphate receptor with a litac molecule. And so we got a map of vacuolar membrane proteins, proteins that maintain the pH gradients that drive endocytosis, proteins that mediate vesicle tethering that are important for vesicle maturation as endosomes mature into lysosomes. But this exocyst complex hit, which is sort of one of our stronger hits from the screen, was really intriguing to us. And so we did a little bit of a deeper dive to understand what role this might be playing in internalization of litacs, or at least mannose-6-phosphate based litacs. And so we knocked down the exocyst complex, and what we observed was a significant decrease in the surface presentation of the mannose-6-phosphate receptor. Well, we saw no change in the cellular levels of the mannose-6-phosphate receptor, suggesting that the exocyst complex or the exocyst proteins were in part responsible for the surface presentation of the mannose-6-phosphate receptor. And furthermore, when we looked at if this complex was maybe just regulating global levels of surface presentation of proteins by comparing loss of surface presentation of the mannose-6-phosphate receptor compared to EGFR, we observed no change in EGFR upon knockdown of the exocyst complex, but again, significant change in the surface presentation of the mannose-6-phosphate receptor. And so what we found actually was a key component of the mannose-6-phosphate receptor's surface presentation biology that had been missing, which was that the exocyst complex is a vesicle tethering complex responsible for binding mannose-6-phosphate receptor rich endosomes to the plasma membrane, allowing them to be then presented back on the cell surface. And so this is actually a key component of the recycling biology of the mannose-6-phosphate receptor that we were able to discover using litacs as a lens to understand this process. But we also wanted to show that litac molecules could be general for uptake of any soluble protein. And so we looked to assemble some pretty modular systems for testing uptake of sort of any litac-based molecule into cells. And so one of these simple systems is to take recombinant MCherry protein an antibody against MCherry, and then an antibody that we've transformed into a LITAC molecule, which is a secondary against the host MCherry antibody. And then we look to see if we can then bring MCherry into cells. And so this process works very well. You get highly specific uptake only with the LITAC tagged antibody. And you get very significant uptake of MCherry proteins into cells across a number of different cell lines. And so this is a relatively rapid way of testing whether or not the receptor that you're targeting as a lysosome targeting receptor is going to bring in cargo quickly. But we also wanted to show that not only were we driving proteins into cells, we were in fact degrading them once they got into cells. And so we chose to look at apolipoprotein E4 as an intriguing potential therapeutic target for this approach, an intriguing soluble target. That's because the ApoE4 allele is a major genetic risk factor for Alzheimer's disease, but also that there have been some interesting preclinical studies suggesting that clearance of ApoE4 from the brain actually allows more ready breakdown of amyloid beta aggregate clearance. So we constructed a LITAC system where we again took a antibody against ApoE4, and then we used our sort of master LITAC secondary antibody to see if we could bring in a fluorescently tagged version of ApoE4 into cells. And so what you can see here is kind of a time course on the right of uptake of fluorescently labeled soluble protein into cells over time. Again, kinetics are very consistent with receptor recycling being sort of a rate determining step in this process. And that you can see actually that you reach this plateau point where you've actually now totally depleted the soluble protein from the media. But uptake does not equal degradation. And so we needed to come up with a system that allowed us to assess not only were we bringing proteins into cells, were we also degrading them when they got inside. And so we looked at a number of different inhibitors of the endocytic pathway, but these can all change actually the kinetics of how receptors recycle in and of themselves. And so things that change the pH of the endosomal system actually aren't great for differentiating between uptake and degradation. But proteasomal inhibitors and sort of lysosome protease inhibitors are great for this purpose because they don't impact the endocytic recycling activity, just the activity of lysosome hydrolases. And so I think about this in a way of when you want to study if your sink is working or not, you can always clog it and see if things build up. And so we found that lupeptin actually did not perturb the ability for us to bring in fluorescently labeled protein into cells, but it did allow us to study the clearance of that protein. And so without lupeptin, you can see that in both LITAC treated as well as control molecule treated cells, you see the same steady state level of protein. And that's because clearance within the lysosome is not the rate determining step. And you're only ever going to see the cellular process sort of at its rate determining step. And so here, what that means is that when you clog lysosomal hydrolases, now you'll see a buildup of protein. And that is exactly what we see. So when we treat lupeptin, we see buildup of intracellular fluorescence of ApoE4 tagged molecules. And this tells us that we are in fact not only bringing cargo into cells, we're also degrading it. So fairly quickly, I think we established that sort of any soluble protein approach was going to be viable, provided some considerations on the sort of, potential biophysical constraints of a soluble target. But I think the more interesting question is, can you drive membrane proteins to lysosomes and degrade them? And again, this is because you ask a fundamentally different thing, which is, can you use a lysosome targeting receptor to override the inherent biology and the inherent recycling of another cell surface protein? So our initial approach to looking at this question was to use sort of a well-validated therapeutic target, but one that also had readily accessible sort of binders that we could easily tap into and use as the initial binding moiety for a LITAC. And so EGFR was a very clear candidate. And that's not only because sort of inhibiting EGFR has been sort of incredibly transformative for a number of different cancers, It's also because EGFR, in its role as an oncogenic driver, also has a number of scaffolding functions. So even when you remove its ability to homodimerize and autophosphorylate, it's also capable of nucleating other sort of heterodimeric complexes and phosphorylation events across a number of other cell surface receptors. And so actually, as a candidate for targeted protein degradation, EGFR makes a lot of sense. And this has also been intensely pursued in the PROTAC world as well. We wanted to show that we could do it from the other side of the cell, because I think this offers some unique advantages. And so what we did was we transformed one of the clinically FDA-approved antibodies against EGFR called cetuximab into a LITAC molecule. So we functionalized it with our LITAC handle. And we asked the question, when we do this simple conjugation reaction, would this transformed antibody act as a degrader now rather than a binder or an inhibitor? And so to study this, we generated two cell lines. One is a cell line expressing a control guide RNA, and one cell line that knocks down the mannose-6-phosphate receptor. And when we treated these cells with our LITAC molecule, so that's the Western blot data shown here on the left, what we saw was fairly robust degradation of EGFR. And so just walking you through this data a little bit, on the left is just no treatment. Far left, in the middle is EGF treatment, and EGF is a known downregulator of EGFR, provided there are no constitutively activating mutations in EGFR. And so we use that as a positive control to study EGFR downregulation. And when you treat with cetuximab alone, you can see that there's not much appreciable degradation of EGFR. And this in part has to do with the fact that EGFR readily recycles from early endosomes. But when you treat with the LITAC molecule, now you can see that you've sort of overturned this recycling behavior and you are now degrading EGFR. So this is incredibly exciting. And we wanted to show that this was in fact due to our direct coupling of EGFR's trafficking to mannose-6-phosphate receptor. And so to do that, when we knock down the mannose-6-phosphate receptor, we see a complete abrogation of the ability of a LITAC molecule to degrade EGFR. So this is very exciting. This suggests that we are in fact using a highly specific lysosome targeting receptor uptake pathway to remove another cell surface protein, that we've overridden EGFR's biology. But there was 20% of EGFR left. And so a common question, I think, is how much of a protein do you need to degrade? What is the maximum amount and what is the minimum amount? How promiscuous is your degradation going to be in terms of your biological effect? And so when we looked at that residual 20% of EGFR that was left in cells, what we observed was in untreated and cetuximab treated cells, EGFR remains primarily at the plasma membrane. But when in LITAC treated cells, EGFR is now sort of concentrated in a number of periplasmic or perinuclear vesicles that sort of overlay exactly with acidified compartments of the cell. And so that 20% residual EGFR in part is sort of stuck in the endosomal system on its way to a lysosome to be degraded. And so here it suggests that a rate limiting step in this process is maybe not sequestration and internalization, but actually maturation of EGFR-enriched vesicles to lysosomes. And so to kind of further show that the ligands that we were designing was driving this behavior, we performed these degradation experiments in the presence of a large excess of monomeric mannose-6-phosphate. Again, these experiments showed us that this inhibited the ability of our LITAC molecules to degrade a cell surface protein. And so not only have we shown that it goes to the specific receptor that we were targeting, it also goes to the specific ligand receptor interaction that we built into our LITAC molecule. And the good news for us is that there's not a lot of soluble mannose-6-phosphate floating around the body. So this inhibitory pathway really doesn't exist endogenously. And then finally, just to show that this removal of EGFR was due to inhibition of the endosomal system, we sort of treat with an inhibitor of endocytosis. Now you can see that this totally prevents EGF-regulated downregulation or EGF-driven downregulation, but also LITAC-mediated downregulation. And so we wanted to look at the functional effects of degradation, even in the context of a known inhibitor. And so we treated cells either with cetuximab or cetuximab Lytac, and then stimulated them with EGF and asked the question, how would phosphorylation levels of EGFR change as a function of degradation versus inhibition? And so when we did that in our sort of control cell line, or our wild type mannose-6-phosphate expressing cell line, you can see that there's robust phosphorylation of EGFR upon stimulation with EGF. And that over time this fades, and this is consistent with a early sort of pulse of EGF stimulation followed by downregulation of the receptor. But when you treat with just cetuximab and then stimulate with EGF, you now see this low level of competitive stimulation rather than sort of a complete loss of EGFR phosphorylation. And this is actually a problem because this low level of sustained phosphorylation is enough to induce downstream kinase cascades. But when you treat with a Lytac molecule now, because you are reducing the steady state of the receptor in and of itself, you sort of significantly alter this steady state level of competitive EGF stimulation. And so here we see actually a functional benefit of degradation over inhibition by itself. And all these effects are lost when you remove the mannose-6-phosphate receptor. We wanted to assess how selective this process might be, because if you're using one of the advantages of going from the extracellular space is that you can readily use highly specific binders, highly specific engineered protein binders to degrade your protein of interest. And so we performed quantitative proteomics upon Lytac treatment to assess what proteins were being degraded. And so in this data, in the upper left-hand quadrant would be proteins that are downregulated as a result of the treatment. And in the upper right quadrant would be proteins that are upregulated. And so when we treated with cetuximab, what we observed is there was really no change in EGFR levels, very consistent with our Western blot data before. And there was some sort of minor changes in terms of some other proteins, but nothing substantial. But when we treated with the Lytac molecule, now you see significant downregulation of EGFR fairly selectively. But there are also a few more hits in this downregulation space, as well as in this upregulation space. And so when we sort of looked in on those, we were kind of asking the question, well, I wonder if we're actually using this as a way to examine a different type of interactome of EGFR by itself. Because you might imagine that a protein that's interacting with EGFR at the post-translational level might also be downregulated upon removal of EGFR. And so, for example, we hypothesized that CD99, which is a sort of serine-rich cell surface glycoprotein that might have a large number of O-glycosylation sites, might actually be an interactor of EGFR. And conversely, when you look at differentially upregulated proteins, these might be ways that the cell is in fact fighting back against EGFR downregulation. And so I think using these quantitative proteomic studies upon Lytac-mediated degradation actually allows us to look at some sort of interesting information about EGFR's sort of native biology, as well as its potential in a disease context in terms of how a cell might fight back against degradation. We've gone on to show this for a number of different targets. And so one of those is PD-L1. And this is exciting not only because PD-L1 is a driver of immune evasion in a number of different tumors and has sort of had a transformative impact in terms of therapeutics that target inhibition of the PD-1, PD-L1 immunoinhibitory axis. It's also exciting from the sort of the fundamental biological level, because PD-L1 has a dedicated chaperone that its job is to recycle it back to the cell surface. And so that protein is called CMT-M6. And so the question for us really was, could we overcome CMT-M6 mediated recycling of PD-L1 from early endosomes back to the plasma membrane? So would a Lytac bypass not only sort of the canonical dileucine motif recycling events, but also would it overcome dedicated cellular machinery? And initially, it wasn't great. Initially, we were using sort of a commercially available, relatively inexpensive PD-L1 antibody, and we turned it into a Lytac as we had previously, and we observed very little surface removal of PD-L1, so about 20%. And by blot, I would say we were in the same regime of only removing about 20 to 30% of PDL1. But when we went back and we asked the question, well, maybe, maybe there's some receptor expression level differences. It turned out the cells that we were looking at these initial studies on, MDA MB231 cells, have a relatively low expression of the mannose-6-phosphate receptor. And so we looked at another cell line that had slightly higher expression of the mannose-6-phosphate receptor. We now saw that there was a little bit more degradation, but I would still say not what we were seeing for EGFR and a number of our other targets, which is kind of about 80% loss of the target protein. And so we went and we re-examined how we were building our LITAC molecules. So originally we were using this commercially available inexpensive antibody. We went and we turned Tecentriq, or atezolizumab, the commercially or the FDA approved anti-PD-L1 antibody, into a LITAC molecule. And now we were able to see robust levels of degradation in these cell lines. We were now degrading sort of the 70 to 80% that we had previously for other LITAC cases. And so this was exciting, and I think showed us that our target scope was going to be a little bit more general than just EGFR. And we were also able to show that this degradation was inhibited by a large excess of mannose-6-phosphate. So again, that it was going through our direct coupling of the mannose-6-phosphate receptor to the degradation of PD-L1. And so we sort of had, I think, kind of dug into a little bit that maybe there was going to be some generality in terms of our target scope of membrane proteins. But we wanted to show that this idea of degradation from the extracellular space was general. And one way to do that would be to go after alternative cell receptors, alternative lysosome targeting receptors that might confer a specific advantage to using them over the mannose-6-phosphate receptor. And so this is a question that Green On, a graduate student that joined me in the LITAC project while I was in the Protosi lab, really was excited about. And so when we looked at the potential scope of other lysosome targeting receptors, one that really stands out is the acylglycoprotein receptor, which has been highly successfully targeted with a number of other directed delivery approaches, including with exciting siRNA delivery approaches. We wanted to ask the question, if we use this receptor, would it allow us to do cell type restricted degradation? Would it allow us to do cell selective degradation? And it would function in a way similar to the mannose-6-phosphate receptor, where it would allow us, where it itself would recycle and not get degraded, but allow us to deposit cargo in lysosomes for degradation. And a great advantage of targeting the acylglycoprotein receptor, there's not a lot of innovation that has to go into developing ligands for it, because the ligands are already well known and established. And so they're accessible with a little bit of synthetic pain, but not too much. And so through a lot of development work, we were able to show that the acylglycoprotein receptor functions very similar to the mannose-6-phosphate receptor in our ability to use it as a lysosome targeter. And so antibody molecules that are programmed with these acylglycoprotein receptor ligand, this tri-GalNAc motif, are able to degrade EGFR in a number of different hepatocellular carcinoma cell lines that express the acylglycoprotein receptor. And that it does it in a way that's almost exactly as efficient as the mannose-6-phosphate receptor. And I think this is really interesting because it suggests that even though there might be expression level differences in the lysosome targeting receptor, and there certainly are for these two receptors, that is not the differentiating factor in terms of what governs how quickly or how efficient the degradation process happens. And so I think as a testament to how exciting this idea is, a number of other approaches have been developed that have also used bifunctional degrader molecules targeting the acylglycoprotein receptor. So both Wei-Ping Tang lab at the University of Wisconsin, as well as David Spiegel's lab, have some exciting results in terms of targeting the acylglycoprotein receptor as a lysosome targeting receptor for degradation. We really wanted to dig in on the question, though, of could we use it for selective protein removal only from certain cell types? And I think this is one of the major advantages of targeted degradation from the extracellular space, is our ability to dial in the degradation capacity from only certain cells, maybe only certain disease cells, for example. And so we conducted a mixed cell experiment where we took a hepatocellular carcinoma cell line, so HEP3B cells that express the acylglycoprotein receptor, EGFR, and the mannose-6-phosphate receptor. And we mix them with HeLa cells that are acyl-glycoprotein receptor negative, but also have EGFR and the mannose-6-phosphate receptor to similar levels as Hep3B cells. And we asked, would we see selective degradation? And we do, in fact, see cell-restricted protein degradation when we target certain receptors. So this is flow cytometry data that shows that we selectively remove EGFR when we treat with the GalNAc LITAC from the hepatocellular carcinoma lines. And that's here, and that's demonstrated here. So GFP, so HeLa cells are expressing GFP, Hep3B cells are not. So HeLa cells are in the right quadrants. Hep3B cells are in the left quadrants. And what you can see is that when you treat with this cetuximab GalNAc LITAC, that you see removal, you see a lowering of this upper left population to the lower left quadrant. But you don't really see a change in the upper right population. But when you treat with the mannose-6-phosphate LITAC, which can degrade in both cells sort of indiscriminately, you see now a lowering of both populations. And so that's quantified here on the right, where you can see that we've demonstrated that one of the major advantages of using the acyl-glycoprotein receptor is that you can degrade proteins only from liver cells. And I think a question when you're driving proteins to some degradation machinery is, do you change how that cell's degradation machinery operates? Are you deleteriously impacting sort of other features of that cell, for example? For us, that question would be, are you damaging lysosomes? And so you can look at a number of different lysosomal markers to ask this question. So one of those is ALEX, which is a marker of sort of when lysosomal membranes get perturbed and disrupted. This is a protein that helps recruit the escort complex to these disrupted membranes to dispose of these lysosomes in a sort of lysophagy pathway. And you can see that when you treat with a LITAC molecule, you really don't see a lot of this pink signal that's in the middle panel here, especially compared with the sort of control lysosomal disruptor molecule. So we're not impacting lysosomes by driving proteins to them for degradation. And we're also not changing the degradation capacity of the proteases within the lysosomes. And that's observed in magic red staining, where I'm showing the quantification of that data here, where you can see that untreated cells, antibody-treated cells, and LITAC-treated cells do not really change their ability for cathepsins, one of the major classes of lysosomal proteases, does not change the ability of cathepsins to degrade proteins. And so we got excited to look at opportunities where we might see a differential effect between inhibiting and degrading extracellular proteins. And so in a collaboration with Jennifer Cochran's lab between Green and Caitlin Miller, we transformed these small integrin binding peptides called gnotins into Knotten LITACs by a simple conjugation to one ligand, one acyl-glycoprotein receptor ligand. And we got some results that were, I think, really intriguing. And one of those results was we actually don't degrade a lot of integrins. We degrade some, 30 to 40%. But the Knotten peptide by itself is also a relatively capable degrader for some classes of these integrin ligands. But when you look at the anti-proliferative effects, here we saw something that I think was really exciting, and that was even in this sort of differential regime of degrading integrins 20 to 30%, our LITAC molecules were providing a significantly more pronounced anti-proliferative effect compared to just the Knottens by themselves, and that's shown here on the right. And so I think this is exciting because it brings up a number of questions, which is how much of a protein do you need to degrade to see an effect? And that degradation does, in fact, in the extracellular space, have an added benefit over inhibition. And so because we had looked at sort of smaller peptide binders, we wanted to ask how much smaller could we get? And we wanted to really develop a modular tool to look at this. And so what we did was we developed FKBP LITACs, essentially. Bumped FKBP variants that we could use a single ligand LITAC lysosome internalizing receptor ligand. and potentially be able to study a number of different proteins and their internalizations and how they might sort of function upon forced internalization. And so as a proof of principle for that, what we did was we just expressed MCherry fused to FKBP and showed that we could internalize this MCherry protein to a very significant degree in hepatocellular carcinoma cell lines using these sort of small LITAC variants. And so we think this could be a very exciting tool for studying a number of different proteins in terms of downregulating them. But sort of one of the, I think, exciting features of this space is that we really don't understand the scope of lysosome targeting receptors. So there are some molecules we know that could potentially be targeted as lysosome targeting receptors that have restricted expression that we might be able to hijack for specific therapeutic contexts. And so I'm showing early data here for one of those that we've identified, which is an overexpressed receptor in the brain and in the central nervous system that might be co-opted for sort of targeted degradation only within those compartments. And so what we did in these experiments is we took our quick MCherry uptake assays, and we expressed actually a genetically encoded LITAC tag that allows us to transform a nanobody against MCherry into a LITAC through simple protein expression that targets this specific receptor that's overexpressed in the brain. And so this early data, I think, is exciting and shows that we are bringing in sort of extracellular proteins through this upregulated receptor, and that protein is indeed trafficking highly specifically to lysosomes. So that's shown here on the right. But this sort of begs an even broader question, which is, okay, so there are some hints about what we might be able to use as a lysosome targeting receptor that are waiting to be liganded. What about trying to understand what is the whole scope of lysosome targeting receptors all throughout the body? And I think this is a really important question, because when you boil down what we're trying to do, we're trying to understand and manipulate communication between the extracellular space and lysosomes. And so it turns out that sort of this is a relatively complex area of biology in terms of endocytic regulation and how molecules from the extracellular space communicate with lysosomes and end up in lysosomes. And at its heart, LITAC molecules take advantage of the ability of one molecule to traffic faster to lysosomal compartments than another cell surface protein. And so understanding not only what proteins can move to lysosomes, but how fast they move would open up a whole space of lysosome targeting receptors throughout the body, and especially in a number of disease contexts. And so that's a question that we're really excited to go after. And so I think understanding sort of what's on the cell surface is certainly an extremely intense area of interest, and understanding the surfaceome. And similarly, understanding what's in the lysosome is an intense area of interest. But approaches that allow us to understand the communication between those spaces have been lacking. And so that's one of the things that we've been working on, which is in collaboration with Manther Abu-Remele, my colleague here at Stanford, we've been using a technique called lyso-IP to try to globally map all of the lysosome targeting receptors in the mouse. And so with this approach, what we're able to do is able to take specifically epitope tagged lysosomes, pull them down and isolate them from tissues all throughout the animal and assess the proteins that might be present in them at any given time. And so when you couple this with a labeling strategy that allows you to label extracellular proteins and study what gets into lysosomes, this allows you to map selective movement of proteins from the extracellular space to lysosomes, and so you can discover lysosome targeting receptors. And this is early Western blot data that shows the viability of this approach. And so what I've highlighted in red across these 4 different organs are all bands, all selective bands that crop up only upon lysosome immunoprecipitation and understanding what's in them. So these are all proteins that selectively move from the cell surface and the extracellular space. So these are also secretor proteins that move into lysosomes and have some presence in lysosomes. And so we can also do this over time. And so this is a project that's being led in my lab by Stephanie Robinson and in collaboration with Greenon and Carolyn Bertozzi's lab. So this is exciting. I think begins to open the door even more to understanding how we might be able to rewire the extracellular interactome for therapeutic benefit. And so we've started to do that with LITAC molecules, and there have been another number of other approaches that have emerged since LITACs to think about manipulating the extracellular interactome. And so other exciting approaches that allow you to bring two proteins together and maybe accomplish some sort of effector function. In the case of these sort of bifunctional antibodies from Jim Wells's lab that allow you to downregulate a membrane protein through proximity to a E3 ligase. There have been some exciting approaches in manipulating the phosphorylation states of a transmembrane protein through extracellular binding interactions. And an area that I think is additionally exciting is thinking about how we might be able to manipulate the extracellular interaction space to remove post-translational modifications from outside the cell. So in the case of removing glycans, I think many of these approaches, I think, are going to have an immense therapeutic benefit moving forwards. And so in terms of, where is potential extracellular protein degradation moving, I think I've given some hints about cell and compartment specific targeting that I think is a challenge both at the basic biology level as well as the sort of molecular design and development level. The development of multi-turnover degraders from the extracellular space, I think, is a key challenge, and one that I think there are a number of really exciting approaches that we'll be able to use to overcome. And sort of the design challenges really feed into where are the therapeutic opportunities for targeted degradation from the extracellular space. And so when we can degrade things selectively in certain spaces, and when we can use multi-turnover degraders in the extracellular space, this really broadens the potential for therapeutic advantages of LITAC molecules or extracellular degraders. And then finally, I think developing a better mechanistic understanding of how we're manipulating endocytosis is extremely important. And so coupling our ability to downregulate a membrane protein with fundamental studies of how this happens and how this competition between two proteins is operating, I think will shed light on a number of really interesting questions, both translationally as well as at the fundamental endocytic biology level. And so with that, I am really grateful to the three extremely brave individuals who've joined my lab so far. Actually, none of which work on LITAC projects. They all have very exciting projects in other directions, as well as I've been extremely lucky to have a number of really talented rotation students and summer rotation students during the past year. And one of the great things about Stanford is the range of collaborators and the expertise. So I owe a big debt of gratitude to Carolyn Bertozzi, Monther Abu Remele, and Green On for driving a lot of the generalization and really establishment of LITACs as a general concept. And especially thank you to the organizing committee again, and I'm happy to take any questions.